Welcome to our poster presentation. Attentional selection can be understood as the process of doing visual categorizations of the form object X has feature I. Something like this object has feature triangle, or these triangles have different colors. We can instruct an observer to select the feature color by asking them to name each of the object's color. In neural terms, feature selection increases the level of activation of cortical neurons coding for that feature, so the object containing that feature will elicit higher firing rates. The level of activation or firing rate determines the speed of visual processing or the rate at which a particular visual categorization is encoded into visual short-term memory. The neural theory of visual attention proposes that the firing rate increase given by a particular feature selection and that determines visual processing speed occurs in cortical visual areas. However, the neural TVA posits that feature selection values do not originate directly there, but derive from frontal and parietal cortex and the limbic system. In line with this proposal, a previous empirical study showed that visual processing speed is associated with the functional connectivity within the cingulopercular network and between the cingulopercular network and the frontoparietal network. The feature selection values that determine visual processing speed must be transmitted from higher order areas to the visual system. Therefore, we investigated whether and how the functional connectivity of these higher order networks with visual networks relate to visual processing speed. We measure visual processing speed by using a whole report task in which participants were briefly presented with letters and asked to verbally report all the letters they were fairly certain they had seen. We model participants' responses based on the theory of visual attention, or TVA, which proposes that visual processing follows an exponential distribution. The slope of the curve at the threshold for conscious perception is parameter C, or visual processing speed, and is given in number of letters per second. 48 healthy participants performed the whole report task and underwent resting state fMRI. For the functional connectivity analysis, we used the entire frequency range available in the data by binning the frequencies from the slowest, or slow 5, to the fastest, or slow 2. Then we examined the functional connectivity between networks of interest and examined the association between network functional connectivity and visual processing speed. We identified 5 visual networks across the 4 frequency bins. The connectivity of these networks with the cingulopercular network was overall negative, except for a subcomponent in the insula in slow 3, where it was positive. The strongest connectivity was observed in slow 4, where it was negative, and in slow 3 with the insula subcomponent, where it was positive. The connectivity of the 5 visual networks with the right frontoparietal network was overall negative as well, with the strongest connectivity observed in the slowest frequency bins. For examining the association with visual processing speed, we assessed 5 linear models, one for each visual network, that included the connectivity with the cingulopercular and with the right frontoparietal network across frequency bins as predictors and visual processing speed values as the outcome. No predictor involving the cingulopercular network significantly predicted visual processing speed in any of the 5 models. In contrast, for predictors involving the connectivity within the right frontoparietal network, we found that for 2 models, the connectivity in slope 5 and in slope 2 significantly predicted visual processing speed. In summary, in the present study, we found that the functional connectivity with visual networks was strongest in intermediate frequency bins for the cingulopercular network and in the slowest frequency bins for the right frontoparietal network. Crucially, we found that the connectivity between visual networks and the right frontoparietal network, but not with the cingulopercular network, was significantly associated with visual processing speed in both the slowest and the fastest frequency bins. In light of previous findings, these results suggest that the feature selection values that are generated in regions of the cingulopercular network might be transmitted indirectly to visual areas through right frontoparietal regions. I'd like to end by thanking the co-authors of this work, the funding, and you for your attention.